Okay, thank you all for coming today. We have, I think, a pretty exciting lineup of speakers, and the first session is going to kick off with an outside speaker who's going to talk to us about innate immunity and adaptive immunity and really how it fits into cancer. Additionally, we're going to have a session which comes after this on epigenomics and cancer, and Smita Krishnaswamy has switched with Richard Flavelle, so she'll be talking in this session, but that will, of course, she'll be talking about epigenomics and how it impacts on cancer immunology. So I'd like to first introduce Nick Haining, who's an associate professor at the Dana-Farber and an associate professor at the Broad. Nick has been a real thought leader in recent years in terms of thinking about how adaptive immune cells interact with both cancer and viral infections and the connections between those two. His talk today is called Genetic Screens to Discover Regulators of Tumor Immunity.
Good morning. Thank you very much for the invitation to come and speak at what I think sounds like an absolutely terrific symposium, and obviously could not come at a more exciting time in the field. First of all, my disclosures. I've had the pleasure of working with companies small and large that really have benefited our science enormously in how we think about the therapeutic development of it. So, as Dean Alpern already pointed out, a lot's happened in the last 100 years in terms of cancer therapeutics, from the point at which William Cooley, is there a pointer? That was the one thing I didn't ask. Do you know if there's a pointer? I thought the mouse would work as a pointer, but it apparently doesn't. Okay, thank you. From the point at which William Cooley who is an orthopedic surgeon at the Hospital for Special Surgery on the Upper East Side of New York, found that some of his patients with soft tissue sarcomas seemed to do surprisingly well if they got death-defying infections associated with that, and tested the hypothesis by actually injecting his patients with ground-up pus, basically. The clinical standards weren't particularly good. The follow-up wasn't particularly good, but there were enough anecdotal reports of improvement that it essentially launched the idea that the immune system perhaps could be entrained to improve the outcome of patients with cancer. So if you fast forward 100 years, it has become clear that that is indeed true, and the efficacy of adoptive cellular therapy with CAR T cells or TIL therapy and checkpoint blockade has really proven remarkable, and has really convinced the most hardened skeptics about the role of the immune system in cancer that this is going to be an essential part of how we treat patients. But if we are going to consider immunotherapy to be a foundational part of the cure for cancer, there is still a lot of work to do. So, for instance, being able to understand why T cells become exhausted in the setting of tumors, and the extent to which that limits our ability to use adoptive cell therapy in the setting of solid tumors is a critical problem. And also, as we've heard already this morning, the development of resistance or lack of response to checkpoint blockade is clearly something that we need to understand and improve. So my lab is interested in both of these kinds of problems. As Nick has already said, we've been very interested in understanding enough about the biology of T-cell exhaustion to start to think about how we might use genome engineering in T cells to engineer away T-cell exhaustion. We've also been interested in using functional genomic approaches to identify ways that tumor cells evade the immune system in order to be able to identify mechanisms that could be exploited to improve the response to cancer immunotherapy or to overcome resistance. And really that's going to be the focus of the talk that I'm going to be presenting today. So the unmet need is clear. As we've already heard, it is the majority of patients it is only a minority of patients who actually respond to cancer immunotherapy. Most of them either don't respond in the first place, or thank you very much, or respond only to only to relapse subsequently. Can you actually see that? You can, more or less. And so it occurred to us that one of the primary issues that needs to be addressed is to broaden the armamentarium of immunotherapeutic approaches that we can apply to the treatment of cancer. And that means discovering new targets. So today I'm going to talk about an approach that we've used that we're quite excited about, which is to use a in vivo genetic screening platform in mouse transplantable models of cancer to identify new sensitivities. Oh, I'm a bit stronger. Thank you. To identify sensitivities of tumor cells that make them more susceptible to immune attack. I'm going to use as a sort of proof of principle of this some unpublished data on a particularly interesting target that we've discovered recently, which is an RNA editing enzyme expressed in tumor cells and others called ADAR1. So much of what we know about how the immune system responds to cancer, we've learned from transplantable tumor models. In which a tumor cell line is implanted into a syngeneic immunocompetent animal, where in the absence of any other manipulation, that tumor will grow inexorably until the animal succumbs. And for many years, we've used workhorse tumor cell lines like B16 melanoma to evaluate the effect of immunotherapies on the growth rate of these tumors. Immunotherapies like GVAX, a GM-CSF secreting tumor vaccine that was developed in Glenn Dranoff's lab about 110 years ago, or PD-1 checkpoint blockade, much like we use to treat our patients with. And in some models, if we apply those immunotherapies in an immunocompetent animal, the tumor gets smaller. So it occurred to us that the parallel states of a tumor growing in an immunocompetent animal unchecked and a tumor in which the immune response is actually causing tumor regression provided an opportunity to start thinking about doing genetic screens to look for modifiers of this immune response that either cause resistance to immune attack or conversely sensitize tumors to immune attack. And the conceptual model that we've used is summarized here in this cartoon, where we took advantage of Cas9 CRISPR-mediated gene deletion to perturb gene expression in a pool of tumor cells, such that each tumor cell incorporated a different guide RNA from a pool of lentiviruses encoding guide RNAs, and was therefore deleted for a different gene. One tumor cell, one gene deletion. But of course, each genetic deletion is represented by thousands of independent deletion events in 1,000 different tumor cells, so that we get an accurate representation of the effect of perturbing that gene on the tumor cell's survival in the face of an immune attack. We could then, at the end of the experiment, where we implanted the tumor cells in vivo and treat with an immunotherapy like PD-1 checkpoint blockade, we could sequence the the library and compare it to identify relative excess of guide RNAs, which might have knocked out genes that recapitulate resistance mechanisms that occur in our patients. Or conversely, if we identify guides that are depleted from the pool under immune selective pressure, one might infer that the presence of that gene was advantageous to the tumor cell in that it protected it from immune attack. So this is the actual experiment that we did, to take Cas9 expressing cells, culture them in vitro for about 10 days to allow efficient deletion of genes to take place and the protein products from those genes to decay. We then passaged some of those tumor cells in vitro. This allowed us to determine whether perturbation of a gene non-specifically knocked off the cell. If you delete beta-actin, the cell's not going to survive anymore, and it's going to be gone. That guide is going to be gone from the in vitro conditions, and there's clearly no effect of the immune system there. An aliquot of the library transduced tumors were then implanted into a TCR-alpha knockout animal, which lacks conventional CD4 and CD8 T cells, and is therefore unable to apply immune selection pressure on the tumor, although it obviously does control for the factors associated with the tumor's ability to survive and prosper in the subcutaneous tissues of a mouse as opposed to in plastic. Further aliquots of the tumor are implanted into wild-type immunocompetent animals treated with GVAX, the tumor vaccine, or with GVAX plus anti-PD-1 to provide, in essence, a gradient of immune selection pressure from nothing at all, growing in plastic, through to a wild-type animal treated with the most aggressive immunotherapy regimen. After 14 days, we would harvest tumors in sequence and compare the guide RNA representation in tumors growing under a lot of immune selection pressure, a little bit less, and none at all. So this is actually what the screen looks like, and as you can see, we achieved our desired degree of range of immune selection pressure, shown here by the rates of tumor growth and by the tumor size at the end of the experiment. Tumors, each one of these representing a different tumor in a different animal, in TCR-alpha knockout animals grew fastest. Tumor cells in wild-type animals treated only with GVAX were in the middle, and tumors in wild-type immunocompetent animals where an immune response is applying selection pressure on the tumor because we've treated it with a vaccine and PD-1 checkpoint blockade were the smallest of all. We had transduced these tumor cells with a library of guide RNAs, initially a library targeting a subgenome scale pool that we had curated from the relevant classes of genes that we might be interested in, shown on the right here. We removed essential genes like beta-actin, because there's not much point in studying that if it is incompatible with the survival of the cell under any circumstances, and focused only on genes that were actually expressed by the tumor cells themselves, leaving us with about 2,500 genes with an appropriate number of control non-targeting guides, and those genes were each targeted by 4 guide RNAs in 4 different pools. So there's an awful lot of QC data that I'm not going to show you, but we convinced ourselves that we were able to see qualitatively robust, high-quality screening data. But the first thing we wanted to do was to determine whether we could actually recover from our screen the sorts of genetic, the sort of gene hits that we would expect to be there. And we were reassured to find that indeed we could find known immune evasion molecules like PDL1 in our screen. So this frequency histogram represents the relative enrichment or depletion of all 10,000 guides that we screened in this genetic screen, and their relative enrichment or depletion in each of the 3 pairwise comparisons down here shown below. And this histogram is shown in collapsed form here. So this is the distribution of all of the guides in TCR-alpha knockout versus in vitro. And as you can see, in each of these circumstances, the guides follow sort of essentially a Gaussian distribution, which implies that the vast majority of guides neither helped nor hindered the growth of a tumor cell. Only a very small number of guide RNAs actually were depleted or enriched. If you look at these 4 red hash marks here, these represent the relative position of each of the 4 guides targeting PDL1 and their relative enrichment or depletion in these comparisons. For tumor cells that are growing in an immunodeficient host versus tumor cells that are growing in vitro, the loss of PDL1 clearly has no effect at all. However, if you compare the distribution of PDL1 targeting guides in a tumor growing in an immunocompetent mouse treated with GVAX versus the same set of tumor cells growing in a TCR-alpha knockout animal where there can be no T-cell mediated response, you can start to see that there's a significant depletion of the guides, meaning that we can infer from that the growth and survival of tumor cells lacking PDL1 is substantially hindered when they are growing in immunocompetent animals treated with GVAX. So perhaps a reassuring result. If, however, we look at the extent of depletion of the PDL1 targeting guides in wild-type animals treated with GVAX and PD-1 relative to the immunodeficient controls, however, we see that there is no longer depletion, which at first might sound counterintuitive. This, after all, is the situation I have already told you in which the most rigorous, the most stringent immune selection pressure is applied to the tumor. Well, the answer for that, of course, is the fact that we're using PD-1 therapy as part of the way of applying immune selection pressure. So PDL1 obviously confers an immune evasion phenotype on the tumor by virtue of the fact that it can engage with PD-1 expressed on T cells and inhibit their function. if we are actually blocking PD-1 as part of the immunotherapy strategy, it now no longer matters whether the tumor cell is expressing PD-L1 or not. And this we found very reassuring, which is that we can actually see very specific biological pathway-dependent results from the screen. And together with a large amount of other data that I'm not going to show you, reassured us that we're able to recover the known biologies that we would expect to be there in a screen where we're looking for immune evasion molecules. But the larger question is, what about the unknown biology? What are the sorts of genes that we might not have expected to have recovered, that we wouldn't have known about ahead of time? And this, I think, was where we first realized that this screen actually might be quite a powerful tool, because there's a large number of biologies that, as an immunologist, I'm not sure I would have guessed a priori would have been involved in dictating whether a tumor cell was recognized and killed by the immune response. We saw genes involved in TNF signaling and NF-kappa B activation, shown here in this group here, and deletion of each of these genes was associated with a substantial growth defect in tumor cells treated with immunotherapy, but not in tumor cells growing in a dish or tumor cells growing in the absence of immunotherapy. So this isn't a non-specific defect in growth. This is a very specific synthetic lethal, if you like, with the effect of an immune response. And we saw large numbers of genes in that pathway, and indeed large numbers of genes even in the same protein complex in that pathway, suggesting that we're actually getting an appropriate level of saturation, genetic saturation of relevant biological pathways in the screen. We saw genes involved in antigen processing and presentation, including some that actually had completely paradoxical effects to those that we were expecting. There's some very interesting stories behind there, and I'm happy to discuss those if there's anybody interested in talking about it, but I'm not going to have time to review each of these today. We also found that deletion of genes involved in the inhibition of kinase signaling rendered tumor cells more susceptible, including a particularly interesting phosphatase called PTPN2, which plays a very interesting biology, which we published last year, but I won't, and because it's published, I'm not going to go into that story today. Genes involved in the ubiquitin proteasome pathway, and most perhaps interestingly of all to us at any rate, genes involved in the inhibition of double-stranded RNA sensing. Which leads to an interesting question: why would a tumor cell care about the regulation of double-stranded? Thank you very much. Great. I now have 3 pointers, so no, it's great. It's better than 0. That's perfect. Thank you. Good to have backups. Always good to have backups. So why would a tumor cell care about double-stranded RNA sensing in terms of its susceptibility to PD-1 checkpoint blockade? So where does double-stranded RNA sensing where does double-stranded RNA come from? So obviously one major source is viral pathogens. Many viruses have an obligate stage in their life cycle in which double-stranded RNA is produced, and so over bajillions of years we've developed hair trigger responses to the presence of double-stranded RNA in the cytoplasm of cells, because that often represents a pathogenic state of infection. But double-stranded RNA can also come from the huge tracts of our genome that used to be a virus. So as many of you probably know, the coding regions that encode the protein products of the genes in our genomes represent only a subset of the real estate in the genome. And a large amount of the genome is composed of sort of ancestral representations of previous viral infections that we as a species have had over millions of years, including transposable elements like L1 sequences, and in particular these allo repeats, these short interspersed nuclear elements that are so profuse throughout the genome and so homogeneous in their sequence that they can often be found very close together in inverted repeats. And so when those inverted repeats are transcribed, the inverted repeats can readily form double-stranded RNA species. So these sine or allo repeats are a very prominent source of double-stranded RNA, which can be produced at any time, and it poses a problem for the host, because you have to be able to distinguish between the sort of stuff that isn't an infection, but yet produces double-stranded RNA, with the double-stranded RNA that really is an infection. And so this is one of the pathways by which double-stranded RNA is recognized and triggers an appropriate antiviral response. There are double-stranded RNA sensors like MDA5 and RIG-I, which together with subsequent adapter proteins elicit a transcriptional response that results in the elaboration of, for instance, type 1 interferon, but together with a whole bunch of other chemokines that recruit in the sort of cells that you would want to be in a virally infected tissue. Other RNA sensors include the serine threonine kinase protein kinase R, or PKR, that has actually quite a lot of roles in the cell, but in this particular instance is relevant because it induces a state of transcriptional arrest and apoptosis, which is sort of what you'd want if it was a virally infected cell. You'd want to stop it from dividing and propagating the infectious payload. But the question is then, so if you get double-stranded RNA from your host, from the host's genome, as well as from incoming viral infections, you would probably want a fail-safe mechanism, a negative feedback module, to prevent the unwanted and untoward triggering of this antiviral response in circumstances where it's not needed. And one of those mechanisms is the effect of the gene that's scored in the screen, ADAR, which is an adenosine deaminase that acts on RNA. Now, ADAR catalyzes the deamination of adenosine into inosine. And although the biochemistry is not fully understood, it's thought that A to I editing event destabilizes the double-stranded nature of these double-stranded species, making them less able to bind to their RNA sensors. The importance of ADAR is, I think, indicated by the fact that ADAR1 knockout embryos, or ADAR1 knockout is lethal at the embryonic stage, and those embryos die due to profound evidence of upregulation of interferon signaling. And in fact, there's a human condition called Aicardi-Gouttières syndrome, some kindreds of which have the syndrome because of loss-of-function mutations in ADAR1. And this syndrome is associated with a type 1 interferonopathy that mimics viral infection. So, and in fact, the importance of keeping ADAR as a negative feedback module in lockstep with this RNA sensing is supported by the idea that both the RNA sensors and this negative feedback molecule, ADAR, are all interferon inducible, keeping the expression of the positive and negative members of the pathway in lockstep with one another. So, having found that loss of function of ADAR increases the sensitivity of tumor cells to immunotherapy in a screen, we obviously next wanted to validate that on a single gene level rather than a pool. So we deleted ADAR with a series of different guide RNAs shown here. ADAR has a constitutive form, which is P110, and an interferon-inducible form of P150. And rather conveniently, just because of the placement of the guides relative to an alternative start site, we could delete either both or one of them. I'm only saying that for housekeeping purposes. It turns out that the phenotype is very similar with both of them, but just in case there are ADAR aficionados in the audience, we use both reagents with similar results. So one obvious question was whether deletion of ADAR causes a non-specific effect on the growth and viability of the cells. That appears not to be the case. As you can see, the cell viability of ADAR-deficient B16 tumor cells growing for a week in a dish was identical to control cells in which a control guide RNA had been transduced along with Cas9, suggesting that there's no non-specific effects of ADAR on the growth and viability of tumor cells. When we took those ADAR-deficient tumor cells, rendered ADAR-deficient by targeting either both or only the P150 isoform of ADAR, there's a very modest effect when you implant those tumor cells in NODS-KID gamma mice, which have virtually no functioning immune system, and perhaps only a sort of negligible increase in animal survival. However, if you implant ADAR-deficient tumor cells, which, as you remember, grow perfectly happily in a dish, in a wild-type immunocompetent animal, you can start to see that there's a significant growth disadvantage caused by the loss of ADAR associated with an animal's survival. And if you treat with just a sniff of immunotherapy, PD-1 checkpoint blockade, which in these control animals is associated only with a transient decrease in tumor size, but really no significant effect on the long-term growth and expansion of these tumor cells, you can see that you entirely eradicate the growth of ADAR-deficient tumor cells, suggesting that loss of ADAR profoundly sensitizes tumor cells to the effects of PD-1 checkpoint blockade. So we were obviously interested to understand the immunological mechanisms behind that, because this was a selective phenotype that only occurred in the presence of an intact immune system, and really little to not at all in the absence of an immune response. This shows the immunohistochemistry of untreated ADAR-deficient or control guide RNA transduced B16 tumors that were excised from the flanks of animals after they'd grown for a bit. And as you can see, ADAR-deficient tumor cells have a lot more infiltrating CD8 T cells, shown here by quantification of this IHC stain, than do their wild-type controls. In order to have a better understanding of the really the kind of the flavor of the immune response that's different in ADAR1 null tumors beyond simply the accumulation of more CD8 T cells, we profiled a large number of CD45 infiltrating immune cells in ADAR-deficient or wild-type cells using single-cell RNA sequencing, and using a combination of unsupervised clustering and curated annotation by signature enrichment or lineage gene identification, we could actually apply labels to these clusters of cells, finding populations of myeloid cells, shown here in these sort of purpley, pinky kind of colors up here, as well as populations of effector cells like NK cells and CD8 T cells, shown in the clusters down here. This is a Tisney plot, which is a two-dimensional representation of multiple in complex gene space. I always find these a little hard to interpret. So we visualize the data in a slightly different way. That's actually much more similar to the way immunologists look at density plots. This shows the distribution of cells and their relative frequency in each of these categories in a wild-type tumor that's just been transduced with control guide RNA. There's a predominance of M2 macrophages, myeloid-derived suppressor cells, the sorts of inhibitory myeloid populations that serve to dampen an effective immune response. And there is a detectable population of CD8 T cells, but it's sort of dwarfed by these populations of immunoinhibitory cells. However, in an ADAR-deficient tumor cell, you can see that the immune landscape in a tumor has been entirely reshaped, so that now you see few, if any, M2 macrophages, a smaller representation of MDSCs. Now there's a much bigger representation of activated monocytes, which actually would promote an effective immune response, and a much larger representation of activated proliferating CD8 T cells, as we saw in the immunohistochemistry in the previous slide. And when we look at the individual gene level, we can see across all of the cells in the, and by single-cell analysis, we see marked upregulation of pro-inflammatory genes, CXCL10, CXCL9, for instance, a marked reduction of genes that have a suppressive effect on the immune system, like arginase 1 and NOS2, which promotes the elaboration of nitric oxide. And a significant increase in the expression of T-cell activation and effector genes like granzyme B and interferon gamma. So in addition to these sort of lineage-related shifts in gene expression across the populations of cells in the immune microenvironment, we also saw a marked effect of exposure to interferon. So this shows a gene set enrichment analysis of an interferon alpha or interferon gamma response gene set in immune cells in an ADAR-deficient tumor relative to a wild-type tumor. And as you can see, there's a highly significant enrichment of these interferon response genes in the tumors. And that is associated with an increased abundance of type 1 interferon beta, as well as type 2 interferons, interferon gamma, in mushed up tumors that lack ADAR relative to their wild-type counterparts. So the loss of ADAR is associated with a resculpting of the tumor microenvironment, associated with the influx of lots of inflammatory effector cells and a diminution of the number of myeloid suppressor genes. So what is the mechanism by which ADAR, the loss of ADAR causes such a profound change in the immunological milieu of the cell? So this is the cartoon that I showed you before, and if this is correct, we might expect that in an ADAR deficient cell, we might see evidence of increased abundance of double-stranded RNA. To assess that, we actually deeply sequenced the RNA from ADAR deficient or control cells and actually looked for evidence of A to I editing from which we could infer how much double-stranded RNA was there. Because remember, if you have A to I edits, the double-stranded RNA doesn't form. As you can see here, either in a resting state or in an interferon-stimulated tumor cell, there is substantially less editing that occurs in the absence of ADAR, suggesting that all of this RNA that's normally edited and normally doesn't form double-stranded RNA is available for forming a double-stranded RNA that could potentially trigger these RNA species. So there should be a lot more double-stranded RNA there. But what is the nature of the RNA species that is normally edited? So we were struck by this marked increase in the amount of editing that occurs when cells see interferon. One trivial reason for that is that interferon upregulates ADAR itself. So more enzyme with a fixed amount of substrate, you might see more editing as a result. But we were curious to see whereabouts in the genome the RNA species that were edited were. So we found predominantly that the edits fall in these allorepeats, these short interspersed nuclear elements. The one I told you that composes maybe 10% of our genome and can readily form double-stranded RNA species. That's probably not a surprise. Others have shown that. What we did find, however, was that the localization of these signs, these double-stranded RNA producing sort of ancestral viral elements, was not randomly distributed. So this is the overall representation of signs across the genome and where they fall relative in terms of their intergenic localization versus intronic and 3' UTR in the darker purple there. However, if we look at the signs that are normally edited by ADAR, they are disproportionately represented actually within the gene body itself in the introns of the gene or in the 3' untranscribed region shown here in green. And I've given one representative gene here. You can see these signs in these little purple blobs here are scattered all everywhere. They're in the gene, they're outside the gene, they're everywhere. But the signs that have edits in them are in the 3' untranscribed region or in the intronic region. So that's interesting. But what's even more interesting is the flavor of the genes, the nature of the genes that tend to incorporate edits. So a clue is here already. I've shown chromatin accessibility and RNA sequence pileups from one gene in particular, IRF9, which is an interferon inducible gene. And what we found was that the genes that contained signs that are normally edited was not a random collection of genes. Rather, it was highly enriched for genes that themselves are upregulated by interferon. So we mapped all 7,000 edit sites that we could find to their position in the genome and compared the relative abundance of those edit-containing genes before or after, with or without interferon stimulation. And as you can see, there's a highly significant upregulation of genes that contain signs that are normally edited in interferon response genes. So quite independent of ADAR's role in conferring sensitivity to the immune response, it suggests that actually this might be a signal amplification system, where if a cell sees a suboptimal amount of virus, for instance, enough to trigger a little bit of interferon, there's a mechanism in place to radically amplify the amount of double-stranded RNA to trigger an immune response. And that is the elaboration of interferon response genes containing these signs, these ancestral viral regions that actually increase the abundance of ligand and under interferon-stimulated conditions amplify any signal that the cell might detect as a result of a virus infection. So this was a complete surprise to us and represents perhaps an interesting explanation for why we have so many signs in our genome. It's because they're a signal amplification mechanism for double-stranded RNA. Okay, but that was a sort of a side shoot. What we really wanted to do was understand, okay, so if there is an interferon-mediated amplification of a double-stranded RNA, what happens to all of that ligand if you don't have ADAR around to edit it and stop it from being detected by RNA sensors? If that's true, what we should expect is that we should see a lot of type 1 interferon in ADAR-deficient cells. In control cells, exposure to interferon causes no increase in the amount of interferon produced by the cell. In ADAR-deficient cells, rendered ADAR-deficient by either of these two guide targeting strategies, at baseline, there's no interferon. The ADAR cells do not spontaneously produce interferon. However, if you give them a short pulse of interferon beta or interferon gamma, suddenly they start producing boatloads of it, because we think the ligand that is normally inactivated by ADAR suddenly becomes much more abundant because all those signs in the interferon-inducible genes now suddenly are transcribed, producing a whole ton of ligand. So ADAR-deficient cells are not spontaneous producers. They just have a hair trigger for producing more interferon themselves. And that we would infer may be caused by detection of that double-stranded RNA by RNA sensors like MDA5. If this pathway also works, we would expect that binding of that double-stranded RNA to PKR would induce translational arrest, and that's exactly what we see. Co-culture of ADAR of wild-type cells with any of these cytokines causes a modest defect in growth relative to control guide RNA transduced cells. However, ADAR-deficient cells are much more sensitive to interferon-mediated growth arrest. They grow perfectly happily if you don't trigger them with interferon. However, if you stimulate them with interferon, you get much more double-stranded RNA, and now they undergo translational arrest, presumably by PKR. So I've shown you that these two phenotypes, interferon production and translational arrest, are associated with these pathways. I haven't shown you that they're caused by these pathways. So to answer that question, we did what I guess geneticists would consider a modifier screen, which is that we took ADAR-null tumor cells, which, if cultured in interferon beta for 15 days, will turn up their toes and die. And we transduced these ADAR-deficient tumor cells with a library of guide RNAs targeting every other gene in the genome to ask whether contemporaneous deletion of any other genes suppressed this sensitivity to interferon. And so a hypothetical result would look like here, where the few surviving clones survived because they have an essential gene for this phenotype that has been deleted. And we saw as clear a result as I've ever seen in a genetic screen, which is that the only cells that were surviving were those that had incorporated guide RNAs that targeted any of the genes involved in detecting or transducing a signal from type 1 interferon, so the type 1 interferon receptor, JAK1, STAT1, and IRF9, as well as PKR, the protein kinase R, suggesting that there is exquisite specificity for this translational arrest and apoptosis phenotype on the ability for the cell to upregulate interferon response genes and PKR to transduce that signal that manifests as a transcriptional arrest. So with this approach, we can actually then dissect, using genetic epistasis, the other phenotype as well, which is not just growth inhibition, which I've shown you, but is production of interferon beta. So this is a complicated slide. Let me walk you through it. Doesn't your heart sink when the speaker says this is a complicated slide? Anyway, so this is a complicated slide, though. So, if you delete ADAR, as I've shown you before, the cells become much more sensitive to growth inhibition, where growth inhibition is on the x-axis here, going this direction to interferon beta or interferon gamma. I've shown you this before. If you delete PKR, as is suggested by the previous screen, that growth inhibition goes away. But the cells still produce type 1 interferon. If you delete MDA5, or its adapter protein MAVs, the growth inhibition persists, but now the cells no longer produce type 1 interferon. And in other experiments, we deleted RIG-I, and the deletion of RIG-I has no effect on either growth inhibition related to interferons or the production of interferon beta itself. So this is exclusively an effect that involves MDA5 and MABs and PKR. So that's very satisfying. It confirms the pathways that we thought are involved, but it also actually gives us essentially sort of genetic probes, because cells deleted for combinations of these genes allowed us to deconvolute the role that production of type 1 interferon or the sensitivity to translational arrest plays in the in vivo response to PD-1 checkpoint blockade from ADAR deficient cells. So this is what the results look like of when we take these cells that are deleted for combinations of these genes in vivo. If you delete ADAR by itself, it is more sensitive to immunotherapy with PD-1 checkpoint blockade. I've shown you that now several times. If you delete ADAR and PKR, the cells are still sensitive to immunotherapy, suggesting that activity through this other pathway that results in the production of type 1 interferon is sufficient to confer sensitivity to immunotherapy. If, on the other hand, you delete the other arm, and by deleting MDA5 in an ADAR deficient tumor cell, the cells are still sensitive, meaning that the induction of translational arrest or apoptosis is also sufficient to account for the sensitivity of ADAR deficient cells to immunotherapy. However, if you delete both of these pathways, you restore the phenotype of an ADAR deficient cell back to that which is equivalent of a wild type cell, suggesting that growth inhibition or interferon production is sufficient to mediate the sensitivity to immunotherapy, but both of those pathways, one or both of those pathways, is required to make an ADAR deficient tumor cell sufficient sensitive to immunotherapy. Okay. So this sensitivity phenotype was so profound, it let us ask a question that I thought was a stupid experiment, which just goes to show that you should never listen to your PI. Any of the graduate students in the audience, never listen to your PI. Never listen to your PI for most things. This is an experiment I thought would never work. We were interested to ask whether deletion of ADAR could overcome resistance to immunotherapy caused by mutations that are frequently observed in patients who develop therapeutic resistance. And one of these that has been known for decades is loss of antigen presentation mediated by mutations in beta-2 microglobulin, the gene that is required to stabilize MHC class 1 presentation on the surface and antigen presentation to CD8 T cells. Nick Rostifo first observed this in immunotherapy-treated melanoma patients years ago. This is one representative paper from my colleague Nir Cohen that found a similar thing using tumor genome sequencing of patients who developed resistance to immunotherapy. So we wanted to model beta-2 microglobulin-mediated resistance to immunotherapy to study whether ADAR could overcome it. So we deleted beta-2 microglobulin, and indeed there's no MHC class 1 on the surface of these cells, and we confirmed that these beta-2M deficient cells were invisible to CD8 T cells by culturing them with TCR transgenic cells that recognize an epitope from ovalbumin. And we compared the sensitivity of wild-type cells to beta-2 null cells that expressed ovalbumin. If the cells do not express ovalbumin, they cannot be detected by OT1 cells, as you'd expect. A wild-type cell that expresses ovalbumin is exquisitely sensitive to cytotoxicity from OT1 cells, but a beta-2 microglobulin knockout is completely invisible to CD8 T cells. So we wanted to ask what happens in vivo. So this is in a setting comparing the growth of untreated or treated control tumors or untreated or treated ADAR deficient tumor cells. We first looked at a different immune resistance loss of function, which is JAK1. If you lack JAK1, you can't transduce interferon signaling. JAK1 deficient tumor cells are profoundly resistant to immunotherapy. And if you lose ADAR1, you still are resistant to immunotherapy if you don't have JAK1, which sort of makes sense. I've told you before that there's an obligate requirement for ADAR to see interferon to upregulate those double-stranded RNA species. But what about beta-2 microglobulin? Well, in control tumors, as you can see, the loss of beta-2 microglobulin makes those cells profoundly resistant to immunotherapy. But remarkably enough, ADAR deficient cells retain their sensitivity to immunotherapy mediated by vaccination and PD-1, even though they are invisible to CD8 T cells. Which is a surprising enough finding that we actually sort of poked around in the immune microenvironment of these tumor cells and found an intriguing possibility for why that might be so, which is that if you look at the overall amount of immune infiltrate measured by the fraction of CD45 positive tumor cells in the microenvironment, as I've shown you before, ADAR deficient cells have many more immune cells in them than do control cells. The JAK1 deficient tumors, which are resistant to immunotherapy, do not accumulate more immune cells. But the beta-2 microglobulin tumors in which that lack ADAR still accumulate a larger fraction of immune cells. And those immune cells contain populations of effector cells, like granzyme B positive CD4 cells, which recognize class 2, not class 1, and NK cells, which don't depend on class 1 for recognition of their targets at all, suggesting that even in the absence of CD8-mediated recognition of tumors, alternative effector populations may be able to exert a therapeutic effect. Okay, so what I've shown you in conclusion then is that ADAR functions as a checkpoint that limits double-stranded RNA sensing in tumors, and that its deletion potentiates immunotherapy and may even overcome resistance by enhancing inflammation and creating an enhanced sensitivity to interferon. And the consequences of this, I think, are therapeutically important because it suggests that there is enough innate ligand in a tumor cell to elicit inflammation if the if the checkpoints that prevent sensing of the double-stranded ligand can be removed. And why is this important? Well, there's a great deal of attention as being paid to mechanisms to enhance the inflammation of cold tumors, either by the exogenous application of, say, STING agonists, or the use of epigenetic regulators that de-repress the expression of endogenous retroviruses. Our data would suggest, however, that actually neither of these is strictly necessary because there's plenty of innate ligand in a tumor cell itself if the checkpoints that prevent that double-stranded RNA from being detected can be identified and inhibited. And for this reason, we're very excited about ADAR as a potential therapeutic target. And moreover, it suggests that actually if you render a tumor cell exquisitely sensitive to interferon, that actually that might be one strategy to bypass the therapeutic requirement for CD8 T-cell mediated recognition of tumor cells for therapeutic effect. So with that, I will stop and thank you and thank all the people who have done the work. I was just the emailer in chief. The screening platform was developed by Rob Mancuso when he was a graduate student in my lab. He's now a senior scientist in the Broad half of my lab, based on some sort of core principles that Natalie Collins, a junior faculty in the immunology in the pediatric oncology department, identified. Jeff Ishizuka has been a co-champion of the project with Rob, and many of the smiling faces here you see contributed to the project. I'm, of course, very grateful to collaborators far and wide, as well as the funders who help keep turning the lights on. So with that, I'll stop and I'll be happy to take any questions.